Hi everyone. Thanks so much for coming to my talk this early in the morning. My name is Salim, and I'm a neuroscience PhD candidate as of 4 weeks now in James Fitzgerald's lab at Northwestern. And today I would like to tell you about the project that I've been working on called Ensemble Modeling Framework to predict synapses from optokinetic stimuli in larval zebrafish. And I know this title sounds complicated, and hopefully as we go, it will make more sense. So my primary research interests lie in tying synaptic connectivity with the functional activity of neural networks. And luckily for me, in recent years, systems neuroscientists have provided an unprecedented data that allows to infer the quantitative links between the two. So our collaborators from Japan and Germany, Dr. Kubo and Dr. Bayer and their lab members, are two such systems neuroscience labs. More broadly, they're interested in understanding neural circuits processing visual motion in larval zebrafish. And more specifically, they're interested in the circuit that underlie the optokinetic response, which is also known as a gaze stabilization reflex in response to moving stimuli. So in their 2014 study, they identified pretectum, a brain region that receives retinal projections, playing a key role in binocular integration. In their 2022 study, they functionally imaged activity in pretectum in response to various moving stimuli using calcium indicators. Specifically, they imaged pretectum while showing larval zebrafish nasalward, or towards the nose, and temporalward, or towards the tail, motions on the LED screens. They first showed 4 different stimuli to each eye and then combined them to create counterclockwise, clockwise, forward, and backward motions. And while segmenting the cells, they identified 11 major response types, a classification based on whether or not the neurons showed substantial increase in activity to one of the 8 stimulus conditions. So here's an example of the response type named MONL, or monocular nasalward left, which, as the name suggests, responds every time when the nasalward motion is shown to the left eye, or this green arrow. Here's another response type called FEL, or forward excited by left, which responds to similar stimuli, except when shown backward motion. Oh, sorry, the labels are wrong. Here. The temporal motion shown to the right eye, so this blue arrow, it should be here, not here, seems to inhibit the response, but by itself doesn't drive the cell past its baseline activity. So if you look at the rest of the major response types, you'll also notice that a lot of them are similarly not driven past their baseline activity, which is why in the coming steps, we will think of this network as threshold linear. Finally, in their 2022 study, they took electron microscopy images, or EM images of the brain of the same fish at different depths and then stitched them together, ultimately sharing one of the first fully automated synapse resolution reconstruction of the entire larval zebrafish brain. And so what's so unique and exciting about this data set, and which also served as a motivation for our project, is that they integrated the measured neuronal responses, shown here in green, with the EM dataset shown in gray, and identified reconstructions of the 207 cells belonging to these 11 major response types. So together we'll call this dataset function-linked connectomics, or full connectomics, and it provides a unique opportunity for us to link synaptic connectivity with the functional activity. And so mathematical modeling is necessary to link them. However, given how detailed this dataset is and how vast is the space of possible models, it's not clear how one would go about figuring out what are the modeling principles necessary for mechanistically explaining the neural network computations. Again, luckily for me, in a theoretical neuroscience setting, Fitzgerald Lab is one of the labs that formalized an approach called ensemble modeling that allows to test many model classes at once. And that's the approach I will examine and apply to this full connectomics dataset. So to explain what ensemble modeling is, let's look at the neural network with population sending feed-forward connections here in orange, and then sending those connections to recurrently connected population here in light purple. And imagine that this is a threshold linear network that was shown some stimuli which gave rise to some patterns of activity. Based on the activity patterns, we can find a set of weights, here represented as a matrix with output neurons as columns and input neurons as rows. And where also red means the positive weights and blue means negative weights, that would recreate the original patterns of neural network activity. However, this set of weights might not be unique. You can as well find a different set of weights that will also recreate the original patterns of activity. And in fact, you can keep going and finding more and more of these candidate models. So the crux of the ensemble modeling approach is that instead of looking at a model, we will analyze all of these models belonging to the same model class, like threshold linear recurrent neural networks, at once, and identify consistent features across the ensemble. So in this cartoon, we find consistently appearing positive weights between this specific set of neurons that signals to us that in a neurobiological setting, that there has to exist such a synapse, and it should be excitatory. Similarly, we can make predictions of the existence of the synapse between other pair of neurons, but now inhibitory. I guess it's worth mentioning that the deeper insight that we get from the ensemble modeling is that we can test whether or not the entire model class, like threshold linear networks, or even the whole input-output modeling framework, can provide us with reasonable ways of linking the structure and function in a biological setting. And for the purposes of my project, we will use this ensemble modeling to make testable synaptic and activity predictions and compare them with what we have in the full connectomics dataset. So we'll start by modeling pretectum as a population of recurrently connected neurons that each represent a major response type and receive feed-forward connections from the retina. Unfortunately, the full connectomics dataset does not include retinal responses, so we will approximate the retina as 4 neural populations that correspond to different types of motions. We can model each neuron in such a network using a standard class of threshold linear differential equations used for modeling firing rate equations, and I'm sure you've seen them in many talks at this conference, where in the absence of the input, the output neuron would exponentially decay, and with the input coming from the retina, this first term, and the second term, and the pretectum, it has to go through a threshold linear function first. So we'll notice from the data that the neuronal response traces have these like steady states. And so for this and convenience purposes, we will model both feed-forward and recurrent populations as exhibiting steady states or approaching the steady states after time t equals 0 when the stimuli was shown. So this original set of differential equations is very complicated. It's recurrent. Everything depends on each other. But when we look at the steady states, what happens is that this originally complicated equations, they all basically decompose into n feed-forward equations. In our case, 16. And so now we will find the values of the steady states by averaging the activity of each neuronal response type for a given stimulus duration and approximate the retina and bias with the average activity of the whole network. And although not captured here by differential equations, we will also assume that the self-connections are not allowed. So now all it takes is for us to find the weights and predict the synaptic connectivity, make and test activity predictions, and then we directly compare them with the full connectomics. So how do we do this with ensemble modeling? So to understand how we find the solution space in a threshold linear network, I would like to go over a toy model that will illuminate the important concepts and quantities that we'll use when applying it to a full connectomics dataset. So let's suppose that we have a very simple threshold linear network with just 2 input neurons, x1 and x2. And they send connections to the output neuron y, w1 and w2 respectively. For the stimulus condition 1, we see a pattern of activity that leads to an output neuron responding. And the threshold linear setting, that's equivalent to this equation here. All the weight combinations shown here, lying on the brown line, solve this equation, and given by this condition 1, and we call this a constraint pattern. Now let's say we have a second stimulus condition, for which the neuron does not respond. In a threshold linear setting, that's equivalent to this inequality. So the solution space for this stimulus condition, which we will call semi-constrained, is now this whole shaded green region. And so now the solution to both of these patterns is this deep brown line, that, well, I guess you can still see, where solutions to each of the stimulus conditions intersect. So looking at the solution space, we can already notice that W1 always exists, and it is always negative. So we can already make a prediction that there has to exist such a synapse that is inhibitory in the biological setting. Making predictions for W2 is, however, more tricky, as it can take different signs. So it's reasonable to assume that the weights are not infinite. So we'll look at the various weight bounds, and that will help us to make predictions about the synapse existence. First, we can find the solution with the smallest L2 norm, which we'll call W-min. That helps us to predict the sign of the most optimized solution for the synapse. Second, we can find W critical, which is the smallest weight bound required to make an individual synapse equal to 0. The interpretation of W critical is that if a biological bound was here in this blue dot, then we would have known that the synapse W2 exists and is always positive. And so basically, the larger the quantity of W critical synapse, the more consistently throughout the solution space that synapse exists and takes, in this case, a positive sign. So the extreme case here is that W critical 1, for example, is infinite, as we already noted that it always exists and takes negative sign. So in higher dimensional case, like full connectomics, we recast this problem as a quadratic programming problem with these constrained and semi-constrained dimensions. And we're still working on this, and as we're still waiting for the complete reconstruction of the whole circuit, but we have some preliminary predictions, but not yet definite results, that I would like to go later in the talk. And in the meantime, we can still apply and test robustness of ensemble modeling by making and analyzing activity predictions. So what we're basically doing when making activity predictions is, given the solution space we already computed, we predict whether or not the output neuron responds to a new pattern of input activity. So remember that we can rewrite this equation as just the dot product between this new pattern vector, x, and all the weight vectors that constitute our solution space. All of these vectors that have an angle less than 90 degrees will have a positive projection, or in other words, the neuron responds when all these weights are on the right side of this blue turquoise dotted line. And for the rest, the output neuron does not respond. And notice that the W-min that we computed previously, the orange dot here, lies on the side where the neuron responds. So now, similar to how we found W critical synapse in our structural predictions, we can define W critical pattern as the bound up to which the neuron will respond to a pattern. So here's the turquoise dot for this particular X new pattern vector represents this W critical new quantity. Suppose now that we have a different pattern vector, X nu 2, for which we find a larger W critical pattern, here labeled by a pink dot. And for yet another pattern vector, we find W critical pattern bound in between this turquoise and pink dots. As I mentioned earlier, we don't know where the real biological bound is. So imagine if this W biological bound was somewhere like in between this W critical nu 3 and W critical nu. Then, after ranking this W critical pattern values, we would expect the ensemble modeling to make correct predictions in a row for higher W critical values. And we will test exactly that when applied to full connectomics dataset. So when applying ensemble modeling to make activity predictions, we need to keep in mind that full connectomics have a fixed number of stimulus conditions, and we can't introduce new patterns retroactively. So what we're going to do instead is we'll find one of the activity, we'll hide one of the activity patterns, recompute the solution space, and find the W critical pattern for a given response type. So here is an example of MONR. And so we do this for every held out pattern, and then we rank the W critical pattern values and see if the predictions for a given neuron when those patterns are hidden match the actual predictions. And so when we do that, we see that the first highest W critical pattern predictions, the first four, are turned out to be correct. Here shown as in green colors. And we get the first incorrect prediction here at the fifth W critical value, and we mark this by a cross. So, we perform similar ranking on the whole matrix, and we see that W critical gets 3 to 5 correct predictions in a row. And that's a very cool result, because the probability of this occurring by chance is very low. But if we want to benchmark it further, or benchmark the robustness of the activity predictions made by ensemble modeling, we want to compare it with the predictions from some model. And in our case, it will be the best fit model. And so to make an apple-to-apple comparison, we need to somehow rank our best fit model predictions and see how well it does as a function of ranking. So we will rank our predictions based on the absolute values of the predicted responses before passing them through the threshold linearity, which will account for the cases where neuron responds positive and doesn't negative in a threshold linear setting. We rank and check the predictions for each response type, and we find that the best fit model, shown here on the right, predicts 1 to 3 correct responses per response type, which is worse than W critical based predictions. So this shows that ensemble modeling approach is robust in making the activity predictions. So as I mentioned earlier, we're still waiting for a complete reconstruction of the data set. I talked about how we have the traces for 11 major response types, but there's still many other response types, and one can go about it with like different classification schemes. So now that's why we're don't have the concrete results for our synaptic predictions, but we still made some preliminary structural predictions at the major response type level, and we found that some of these predictions seem biologically plausible. For example, highly ranked predictions from the ensemble modeling include excitatory connections from retina to monocular response types, which is highlighted here by this orange ellipse, and that is in tune with what we know about the retinal projections. It also predicts connections from monocular to binocular response types and recurrent connections between response types that respond to similar stimuli. More excitingly, we predict the network to be mostly inhibitory, which is also in line with some of the new experimental findings from our collaborators in Japan. And so, but I said it earlier, we still don't know, we're still waiting for the complete reconstructions, so we don't know yet whether or not the framework would be able to make accurate synaptic predictions. But eventually, we want to make predictions at the level of single cells, and we might also realize that this level of abstraction might not be the most appropriate, so we'll have to find some intermediate level where the ensemble modeling performs the best. And in that regard, we've been trying different classification schemes and making predictions with more response types. And I would love to share more of my findings at future NITMB events. And so just to summarize, full connectomics dataset provides us with a unique opportunity to link structure with function. Ensemble modeling approach, formalized in a setting of threshold linear networks, allows us to rigorously test these links between the structure and function. And ensemble modeling makes robust activity predictions and interesting structural predictions that are yet to be further examined and tested. And I guess with that, I want to thank all members of the lab and our collaborators, and all of you for coming to this talk early in the morning. And yeah, that was my first ever conference talk. Thank you.
So Salim, that's fantastic. Second-year graduate students speaking to a large conference like this. Any questions?
Hi, great first talk.
Thank you.
So I have a kind of biological question about W critical. When the brain develops during development, right, it's rewired and it changes a lot. So do you think the W critical is also going to be different from different stages of the brain?
Yeah, I think that's a good question. Yeah, I think part of the reason, so I think I guess in general, my answer would be most likely. But for this particular project, I think they looked at the larval zebrafish brain at the time when I think this particular circuit seems to be mostly wired. And so, yeah, but I guess in general, yes, this W critical values would like change as the brain develops, or even as, you know, yeah, connections keep changing.
I may have misunderstood something, but how important was it in terms of in terms of sort of the underlying mathematics, you were looking for solutions sort of in a constrained space, and I'm wondering how important it was that the constraint was actually a straight line versus like how important to this is the threshold linearity versus something sort of generic enough that it's as long as it's a curve that sort of wanders away in the right direction.
Yeah, so I guess we haven't looked at the other transfer functions. This approach is mostly formalized for the threshold linear functions, and the reason is it's like pretty like intuitive and mathematically tractable. But yeah, I guess this would probably change if you have a different network. Yeah.